And so today's paper, it's another old one, but I like it because it's got a microbial diversity angle that I like. From Craig Venter, Environmental Genome Shotgun Sequencing of the Sargasso Sea. It's a science paper. This was one of the first large-scale metagenomic papers that came out. And so a lot of the paper is made up of gee-whizzing you with millions of sequence reads. That's a lot harder to gee-whiz people today than it was then. I don't remember whether I talk about, yeah, okay, so what is the Sargasso Sea? Anybody know where?
In Greece.
Hmm?
Greece.
Nope. Nope. Nope.
Colorado.
It's sometimes called the sea without a shoreline. The Sargasso Sea is a lens of warm water that sits in the middle of the Atlantic Ocean. It's called the Sargasso Sea because sargassum collects there. Sargassum is a kind of seaweed. It sits right there. It's in the northern Atlantic. It's if you think about how the trade winds go, the northern Atlantic circulation, the Sargasso Sea is right in the middle of that. And it's trapped there by that circulation. It's been studied a lot. A lot of early microbiology, a lot of early microbial ecology occurred from studies in the Sargasso Sea. It's easy to get to. The weather's a lot better than other oceanic environments. I mean, you can stop off at Bermuda and just drive your boat out in your boat. Craig Venter was the guy who did this. His group sequenced some of the first microbial genomes, went on to do the human genome. He was one of three groups that did the first human genome sequences. And because he's a complete and utter egomaniac, he went on then to do something even bigger. And so that's metagenomics. Have any of you ever seen Craig Venter in a discussion, TV, something? You'll never forget. He, I don't think, has ever confirmed it, but there is no doubt whatsoever whose genome that was that they did. There's a slideshow that you can go through if you want to see that. It's basically the same talk. What he did, they selected 7 locations. So they drove his yacht out into the middle of the Sargasso Sea and collected seawater from 7 different locations. Well, first of all, how do you collect seawater bacteria? You drop a tube down into the ocean. Typically, it goes down a few meters, so you're not skimming stuff from the surface, but you're not going deep either. And you start to pump water over a tangential flow filter. So this is a filter, it's about so big. And it continuously flows water over the one side, and it's highly pressurized, pushing the liquid through. And it circulates it so that you continuously concentrate the material across that filter. And ultimately, the filter then gets coated in bacteria, and you can scrape that off and do whatever you want. You have to use a pre-filter. Before we go to the sequencing data, let's look at the pre-filter data. So these are like sterilizing filters because you want to collect bacteria. But if you don't use a pre-filter, they get instantly clogged, the first piece of seaweed that gets in, and you block your filter, right? So you use a pre-filter, which is usually a fairly coarse, like Watma paper, something like that, to filter out all the big stuff. And so in this case, they primarily used 80 micron, or excuse me, where's the filter? This is not right. This is filter, pre-filter. I can't. Yeah, I guess it could be. It's an 80 micron. So these are fairly big pre-filters. These ones are bigger still. And so you're filtering out all the essentially eukaryotes, right? At least all of the typical plankton sorts of eukaryotes, algae, larvae, animals, that sort of thing. And then you've got the sterilizing filter to collect all the microbes. Now, they use one here where they're looking for bigger stuff. And you'll see that they got more eukaryotes when they did that. So you scrape off that filter, you isolate DNA, and then you start to sequence. The way they did the sequencing was to clone 3 kb fragments. This was in the days before high throughput sequencing. And then sequence in from each end so that they've got 2 sequence reads that they know are associated with each other. They did essentially 2 million sequence reads. Utterly impressive in the day, not so much today. They got 1.3 billion nucleotides of sequence. Well, excuse me, 1.5 billion. And then these are, because oftentimes if you get more than one sequence read, you can see that they overlap and you can assemble them together. And so they tried to assemble together as much of the sequence as they could get. And these are called contigs. When you can assemble several of them together, these are contiguous sequences, or contigs. Mini scaffolds are just if you get a good sequence read off both ends. I don't know why they include that in the data. And then singletons are where you only get one sequence read off of here. Of this essentially 2 billion nucleotides of sequence, they get about 1 billion unique sequence. 1 billion nucleotides of unique sequence. So half their sequence is duplicates of other things in there. That's not necessarily bad. That your overlap is always going to be a duplicate, for example. They got a bunch of scaffolds. They got, what, just shy of 1,500 ribosomal RNAs when they did this. And the rhodopsin thing, we'll talk about that in a minute. Can you think, so we've talked about metagenomics a little bit in context of pulling out ribosomal sequences by PCR from in the environment. That's a kind of metagenomics as well. Can you think of an advantage of this as opposed to the way we've talked about it so far? Yeah.
Well, if you have an organism that doesn't bind to the PCR.
Right. So when you amplify, what you get depends fundamentally on what your primers have. So you can have bacterial-specific primers, but there are lots of organisms that those primers don't hit. And particularly amongst organisms that you don't know about yet, you can't design those into the primers, right? Because you don't have their sequences. And so what you get from one of those ribosomal metagenomics is typically a lot more of what you've already got. Now, the bacterial primers are pretty good. Eukaryotic primers and archaeal primers stink. For years, people thought that Methanobrevibacter was all through the environment, that people hadn't ever realized that it was everywhere. Well, you look at the primers, and that's the main thing it hits. And the universal archaeal primers are terrible. This eliminates that bias, right? But your sequence reads are about 500 nucleotides long. So you're not getting whole genes, you're getting a chunk of the gene. You can't assemble these. There's so much sequence conservation that if you assemble them, you're almost certainly going to be putting 2 different organism sequences together. We talked about proteorhodopsin. They found a bunch of them. Now we know that SARD-6 is common in the environment. It turns out that many of these sequences are not from gamma proteobacteria or SARD-6. They're able, by blasting the sequences, to assign these to either bacteria or eukaryotes or archaea. And the bacterial fractions, the assembled stuff is pretty high. I'll show you why in a minute. And for most of these, they're, more than half of them, of the sequence they got was bacterial. They did get a fair bit of eukaryotic stuff. This is really interesting. These are organisms that are smaller than a micron in diameter. And it turns out that if you use eukaryotic specific ribosomal probes in the ocean, somewhere between 10 to 40% of what you see are eukaryotes in the 1 to 2 micron size range. They look like bacteria, but they're not. And we don't know very much about them. These are just giant numbers of look at all the sequence we got. Notice that the big filter, the filter with the large pre-filter and a large pore size, collected the most eukaryotes and had the smallest bacterial fraction. Most of the bacteria, of course, slipped right through that filter. So they got a bunch of sequences. Keep in mind that although they were able to assemble up to about half of the sequence, really still a majority of the sequences are single short sequence reads. And so you can translate those, look for an open reading frame through that sequence, translate it, blast it, and see what you get. And they're able then to determine all these numbers for genes they got from different metabolic groups. Cell envelope biosynthesis, cellular processes, I don't know what that is. Central intermediary metabolism, basically the Krebs cycle and associated things. Energy metabolism, so forth and so on. Every one of those genes is patented. Yeah. They own those sequences. Ask me if I like it.
They own genes
Yes, that they fished out of the ocean.
genes for central carbon value.
Yeah.
In general or just for bacteria?
Every sequence they got, they own. They own.
How do you own?
Yeah.
Maybe a star. Wasn't that overturned with the Supreme Court case?
Some of it was. Some of it was. But then someone has to fight it, right? They still got that patent. And so now if you want to use it and not pay the royalties, you have to take them to court and show that the Supreme Court ruling does apply in that case.
How much are the royalties?
I have no idea. And I don't know how many of these have actually been used.
If you own a genome, I guess, would you be able to determine what the royalties would be, or are they like set?
No, you assign those. You come to a legal agreement with the owner of that intellectual property. The folks who own Taq polymerase have made billions of dollars on it in licensing fees and everything else. This is why people like archaeal RNA polymerase or DNA polymerases these days, because what they patented were bacterial DNA polymerase, or excuse me, microbial DNA polymerases within a certain size range, and the archaeal ones are bigger than that. It's silly. So, probably the most abundant organism in the surface ocean water environment is Prochlorococcus. This is a cyanobacterium. We talked a little bit about it earlier. And so you might expect that they would get a lot of Prochlorococcus sequences, and they do. And so the outer circle here is the cultivated Med4 strain of Prochlorococcus, this is the complete genome sequence. All the inner circles then are chunks of Prochlorococcus DNA that they were able to assemble out of their raw data. There's more than one circle because there's more than one strain of Prochlorococcus in the ocean. And so they're able to show that there are at least 5 major strains of Prochlorococcus in the environment, meaning that, so for example, in this region, they've got some differences in sequences or gene order here, here, and here. And likewise, this stuff. And you can show that you can exclude the possibility of there being any less than 5 strains of Prochlorococcus in here. You cannot say what the upper limit might be, because there's nothing to say that this sequence and this one are in the same genome. They could be in 2 different strands, right? And so there could be hundreds or thousands of sequences of strains of Prochlorococcus, but there's at least 5 major ones. This is what the data looks like. So this is the chunk of genome, and then each one of these is a sequence read, and you can see they overlap, and they're able to put them all together. Now, when you do that, their sequences aren't always going to be exactly identical. And so here's the standard strain, and then you see this organism has a CT here instead of a TC. This one has, I don't know what that is, I can't read it. Maybe a deletion. I think that's what that might be. This has a G there instead of a T. These are called microheterogeneities. So there are variations in sequence in just individual nucleotides. Does this sound familiar to you from genetics? Isn't this the way, if this were a eukaryote, wouldn't you call that an allele? So here you have a bunch of organisms with allelic variation in their genomes. This is kind of what you expect, right? So we talked a lot in this class about diversity in terms of, you know, all the different major families and kingdoms and orders of organisms that are present. You know, it's 65% Proprionobacteria. But there's another kind of diversity in microbial communities, intraspecific diversity. So this is diversity within a single species. The oceans are not full of one giant clone of Prococcus. The organisms differ a lot. Where does this variation come from? Ultimately, of course, mutation, right? And those mutations are selected. And just as in eukaryotes, or just as in sexually reproducing eukaryotes, these organisms trade their DNA around, and you get rearranged reassortments of these alleles and selection across populations of the alleles as well. Now that sexual exchange of DNA doesn't occur as part of reproduction because they're bacteria, right? But cells leak DNA, cells lyse, organisms take up DNA, and you might imagine, for example, if this organism took up this piece of DNA from the environment, it could undergo homologous recombination, right? And now you just change the sequence of the genome, and that may be better or worse than it was before, and that selective pressure will drive the population one way or the other, exactly as it does for plants and animals. It's just as in a mammal or tree population, that allelic variation strengthens the population, right? That's diversity in the population. It's just as important in bacteria, and when you look at environments, you always see this kind of thing. If you look, for example, at the outflow of a Yellowstone hot spring, you see a bunch of chloroflexes, but you get the same kind of allelic variation. Some of it is apparently haphazard, like this. Some of it is adaptive. And so if you look down that spring, in the case of Yellowstone, you'll notice that there are different alleles at different temperature bands. Likewise, they don't show it here, but you see the same specific sets of alleles being more abundant at different depths in the water column. So they're adaptive to their local environment.
Do those alleles necessarily help them survive at that depth?
Some do. We'll talk about proteorhodopsin in just a moment. And some maybe are hangers-on or neutral. So Prochlorococcus wasn't the only thing they got big chunks of genome from. They also got essentially the entire genome of Shea-Winella. This is a heterotrophic organism. Here's the genome of the cultivated organism, and they've got two major strains of Shea-Winella in there, at least. Notice that there's some color differences here. What this means is that a chunk of sequence, of course my color vision is not that good, but somewhere in here, this chunk was swapped into the middle of this content. So this is a genomic rearrangement. And you can see that it's different in this organism. Here they can be a little more certain about, these major chunks, for example, we know that it's a different strain depending on whether this piece is actually absent in this one, which that would be over here somewhere. This is presumably a deletion. And this is another graph showing a bunch how these overlap in the contigs, and again, you're seeing microheterogeneity in the sequence. I don't show it here. Maybe it's in the slideshow. Maybe I should do the slideshow part for this. Here's another genome sequence. That's fuzzy, that's okay. They were able to get the essentially complete genome of Burkholderia with zero microheterogeneity, at least within the limits of the of sequencing errors. So that's pretty weird, isn't it? Here's a population of organisms on the surface ocean, abundant enough that you can get the whole genome and no allelic variation to speak of. What's Burkholderia?
That is human pathogen.
It's a human pathogen. They didn't think anything of it, but the folks who've been doing the ecology of the open ocean recognized right away that this was not right somehow. And so, I mean, maybe somebody sick handled the filters. No evidence that any of the people on this yacht had Burkholderia infection. And so they sifted through the data, and this data is a nightmare to search through. We tried to pull out RNA-seq sequences from it and just couldn't do it. The data is in weird formats. It's hard to understand. It's all interlinked in weird ways. It's really problematic. But they were able to search through it, and they recognized an important feature of these sequences, which is every single one of the Burkholderia sequences came from one of the 7 filters. That one filter was the only filter used which wasn't new. And they didn't know where it came from. I'm guessing blood dialysis. All right? That's a ton of data. I don't know how many hundreds of thousands of sequences that is, but it's a lot. And it's meaningless. Well, here we go. Contamination. Just like PCR, this high-throughput sequencing is an incredibly powerful technique, which means that when it goes wrong, it goes powerfully wrong. Keep that in mind. They got a number of plasmids as well. Some of these are quite large. This is 200,000 base pairs. That's a chromosome. All right. How do they divide them? If they got a circle without a ribosomal RNA gene on it, they call it a plasmid. But it's still a chromosome. These are metabolic genes, primary metabolic genes. You see the colors on them? So those are minor chromosomes in the host organisms. And they got a bunch of those sequences. These are sequenced to completion. Some of them were quite small. This is just a handful of genes. They got some viruses. How can you get a virus when your filter isn't that big? Giant ones? I don't know that they got any of those, but they got a bunch of traditional bacteria. Yeah, so the phage-infected bacteria, right? And they got a lot of that. I'm not going to talk. They got a big chunk of an archaeal sequence, but nothing's ever come of that, so I don't talk about it. They got a bunch of proteorhodopsins. We talked about the importance of proteorhodopsins earlier. And they got enough of them that had flanking sequence. So if you get a chunk of sequence right in the middle of the gene, all you know it's a proteorhodopsin. But if you get from the end, you've got some flanking sequence, right? What if you take that flanking sequence and blast GenBank and find out what kind of an organism it might come from? Now you've got an idea about what organism that proteorhodopsin sequence resided in. And when you do that, you find that they fall into these classes, and they are from all of the major phyla of bacteria, except cyanobacteria. So this proteorhodopsin is a little bit like nitrate reductase or NIF genes in that they are mobile and they seem to exist in all different kinds of organisms that can grow by phototrophy. You can look at this tree in great detail if you want to do that. These are the original proteorhodopsins from the Sauroides 6 group. They also see some halorhodopsins. Yeah, they see some fungalopsins too. One of the things that they've worried about a lot is the bias of ribosomal sequences. And so the question is, can we look at this population and kind of sort out who are the major players based on something other than ribosomal, or compare it to ribosomal and see how consistent they are? And so they choose some highly conserved protein coding genes and then ribosomal RNA, and then do the phylogenetic analysis and see what groups they belong to. And alpha proteobacteria are the big ones. And you can see that the numbers they get for these, elongation factor G, TU, heat shock protein 70, REC-A, and RNA polymerase subunit B, pretty consistent, right? Somewhere around 35 or 40% of the organisms were alpha proteobacteria as measured by any of these genes, including the ribosomal RNA. Beta proteobacteria, there's a lot more variation. You see a lot more of them with the ribosomal RNA than you do with these other ones. Gamma proteobacteria is pretty consistent, although you see more of them in the ribosomal than the others, and so forth down the line. Keep in mind that the biases we've talked about already. And so, for example, why might you see a lot more REC-A sequences from Deinococcus thermus group than from any other gene? They've got multiple copies of REC-A. Remember, this is a DNA repair enzyme, and that's what Deinococcus does. And so all of these have this bias. Gamma proteobacteria tend to have high copy numbers of ribosomal RNA, so you might expect that peak to be a little higher. I find this really interesting. Synechococcus is the most abundant organism in this ocean, but as a fraction of the total, it's not really any higher than anything else. What that implies is that this might be a fairly monotonous population, but these must be very highly diverse populations, and they are.
So the higher the bar, the more
this is the fraction of clones that contain that. So in this case, what, 41% of the ribosomal sequences were alpha proteobacteria.
And these are clones that are unique from each other in some way or form.
Unique.
Or
They don't do an analysis breaking those down and see how diverse the individual sequences were. That wasn't part of their interest. All right. So what we're going to do on Friday then is take this to the next step and ask yourself, okay, who's really out there using high throughput metagenomic sequences and focusing in on the question of what's this population really look like and who's out there. One of the things you'll notice is that they don't, if you add up the ribosomals, you pretty much use up all the organisms in these categories. That's one thing they don't even mention in the paper. The implication of this is that most of the organisms, at least in this environment, are from these well-established groups. That there aren't big swaths of phylogenetic groups in the ocean that we don't already know about. That would be an important thing to know, right? The paper we talk about Friday, they argue that there is such a group. I think they're wrong, but I think they're wrong for fun reasons. And I might be wrong. All right, so that's it for today. I will endeavor to get the exam, the take-home part of the exam to you as soon as I can. And I didn't want to do that. No, it's due Monday. That was pushed off as a yeah. 1:30.